Well, good afternoon. Good afternoon, everybody. Welcome to the Muscle Health Research Center's second seminar of the term. We're delighted to welcome this afternoon Dr. Katsu Funai from the University of Utah in Salt Lake City, a beautiful area of the world. Dr. Funai got his bachelor's degree at Boston University and did his master's there also. Worked with Roger Fielding, studying protein metabolism. He then went on to do his PhD at the University of Michigan. And he worked with Greg Carty and studied a lot of glucose transport. And so got an experience studying glucose metabolism. Then he did his postdoc at Washington, University of Washington, Washington University in St. Louis, and worked with Clay Semenkovich and studied fatty acid synthase, fatty acid metabolism. He got an assistant professor position to begin with at East Carolina University, and after 4 years was recruited to the University of Utah, where he is right now. And so we're looking forward to hearing about his work on mitochondria, mitochondrial lipids, and how they regulate metabolic efficiency. So I'd like to invite Katsu to turn on his video and share your slides with us and take it away.
Okay. Thank you, David. Can you see my slide?
Yep.
Great. And thanks for attending this seminar, my talk. Did you want me to turn off my video, or I don't know if that's preferred?
You don't? Okay, sorry.
I'll just leave it on. So I have not been to York, but I have visited Toronto when I used to live in Michigan, like David said. Beautiful city. Really loved it. I've been there actually for my wife's conference and went to Blue Jays game. A lot of good memories. I really love that city. So I can't unfortunately visit in person, but I am very fortunate to be giving you guys talk over Zoom. I think there are a lot of negative things that happened over the pandemic, but perhaps this is a few of the good things. Perhaps it has created some opportunity for us to interact remotely in the ways that we have not had an opportunity to do so. And so I'm grateful for the opportunity for me to present my laboratory's work today to you guys at York, and I think some people may be joining from elsewhere. I see Brian attending. So thanks for being here. So my lab is interested broadly in metabolism. And that's a really broad definition of what our lab is interested. As Dave has described, my research emphasis have been heavily on skeletal muscle. That's been my training, and I identify myself as an exercise physiologist. But I think my interest in metabolism and metabolism skeletal muscle have led me and my group to understand the relevance of what we have found in muscle in other organs also. So if you have looked up my name in PubMed, you may find topics of various pathways and or tissues. But I think where my heart remains to be in skeletal muscle research. That's where my first break has been as a student or a PI, and for sure my training. And I think in particular, because I'm presenting at the Muscle Center at York, I will be focusing my my presentation relevant to our research on skeletal muscle. and mitochondria. Okay, let me make sure I can move my slide forward. So stepping back a little bit, the overarching theme in our laboratories is to understand mechanisms for metabolic diseases. And when we say metabolic disease, there are a few things to keep in mind. And so I think oftentimes we attribute the cause of metabolic disease to obesity because that is one of the most frequent, I think, nowadays reasons or co-manifestation of metabolic diseases is happens in those people who are overweight. We also know, though, that similar pathology could happen in those people who are absent, who are not overweight, people who may be, who may have sedentary lifestyle or who are not able to move, that may or may not be overweight. And they are similar diseases that could happen in the absence of obesity or inactivity in the case of genetic diseases and so forth. So when we think, and when I use the phrase metabolic disorders or disease, we have to be intentional about what do we actually mean, because we tend to confuse causality, correlation of these many different diseases and sum it up. And so we keep that in mind, but still use the phrase metabolic disorder as a convenient terminology to sum up a lot of different things that may happen due to many different causalities. So when we think about, for example, obesity or overweightedness induced metabolic disorders, some of the questions that we try to think is, what is it about, for example, obesity that cause metabolic disorders? Because a person who is obese have increased adiposity, which in itself have cellular and downstream consequences that is manifested systemically. A person who's obese also have altered circulating cytokines and metabolite profile, also may have different dietary composition, increased energy or acute increase in energy intake or acute or systemic decrease in energy utilization. So there are many different reasons why or different things that happen when a person is obese. And the same thing could be said for a decrease in energy utilization or inactivity. When a person is inactive, this person may or may not be overweight or have increased adiposity, may or may not have altered circulating metabolites and cytokines. So it's important to not confuse co-manifestations with causality. And I guess the intro segue to what I'm about to talk about is this concept of energy balance. And that is something that we think is important. One of the things that is important. We're not claiming that it's the only important things, but we clue into the fact that in both obese and inactive condition, there is an energy mismatch that happens very frequently in these individuals. And we think that this imbalance in energy intake and energy utilization may be contributing to metabolic disorders. So what are we talking about? Of many different downstream consequences in different tissues and organs that might happen, I think one that we think is important is bioenergetics in mitochondria. So in weight stable condition, we could presume that the amount of calories that's being consumed approximately equal to the calories that's being expended. And I show a cartoon of a burger and a person running on the right, but of course we know that, you know, physical activity is not the only form of energy expenditure, but we know that that is one fairly robust way to change energy expenditure by being active or not. So in the homeostatic state where there is a balance between energy in and an energy out, we have a mitochondria that has a flow of energy in and out in a relatively unobstructed way. But then what happens when there is an imbalance to this energy in and out relationship? And so this can happen, like I said, with an energy excess or reduced energy utilization. But we believe that when there's a mismatch to energy in and out, that mitochondria adapt. And mitochondria adapt in a way essentially to reduce a redox pressure that is building up from the fact that there's a lot of energy that is coming in, but there is not a lot of energy out. And so over a period of time, an extended, prolonged state in which there is an excess energy and limited energy out, and we think that the mitochondria adapt such that it becomes energy inefficient. And by that, I mean there is a secondary outlet of the energy that gets released so that this pressure that is building up in mitochondria is relieved. And so this energy out is not in the form of ATP resynthesis, and it has many secondary consequences, including counterbalancing energy that is coming into the cell. So it's not necessarily maladaptive, but I think it has consequences that over the period of time can be detrimental to the health of the cell or to the system physiology. So what are we talking about specifically when we say inefficiency and mitochondria? We're talking about the energy transducing processes of oxidative phosphorylation. And there are many ways to think about this. We, at least for now, are thinking about the energy transducing steps of oxidative phosphorylation in this manner. And you can read about this in the review that we published earlier this year. But we break it down into multiple steps. And this is obviously because OXPHOS is not a single reaction completed by a single enzyme, but is a combination of multiple processes that happen. An important point here that I'm going to allude to later is that it's a process or processes that happen through and across in a mitochondrial membrane, which I'll be talking about later. But regardless, without going into too much details, the 4 steps that we define as steps of oxidative phosphorylation is as follows. So the step 1 is simply electron donation by NADH or succinate into complex 1 or 2. And these electrons come from foodstuff, specifically through TCA cycles, beta oxidation, and so forth. So that's step 1. And step 2 is a process by which the electrons that's been donated to complex 1 or 2 are transferred within these complexes and among complexes such as 3 and 4, complex 3 and 4. And these complexes, 1, 3, and 4, utilize energy from these high energy electrons to pump protons that exist in mitochondrial matrix against electrochemical gradient into intramembrane space. So this is, we define step 2, electron transfer. Step 4, so I skipped step 3. Step 4 is what is known as ATP synthesis, where membrane potential that's created by protons that's been pumped into by step 2, and I'll explain step 3 a little later. But this membrane potential generated preceding to step 4 is then utilized as proton motive force to resynthesize ATP. So that's step 4. Step 3 is merely a way for us to make a note that number of protons that's being pumped by step 2 into membrane space does not equal membrane potential for 2 reasons. One is that number of protons that's being pumped in needs to be subtracted by a number of protons that's being leaked out. And I show that here in step 3 by a protein named UCP, or uncoupling protein. It's intentional that I don't say 1, because this UCP symbolizes any given enzyme that we know that exist, or we know there may be others that have this function, but regardless, there are presumably mechanisms by which protons can leak back to matrix. And it's not just the membrane potential is not simply the mathematical sum of protons being pumped in and leaked out, but it's actually, because this membrane potential is actually a delta of concentration of proton, so it's actually need to be divided by the volume of the intramembrane space. And we think that could also be affected. So why are we interested in thinking about OXPHOS and into these steps? And that's because a consequence or consequences of inefficient energy transfer processes in each of these steps would be different. So inefficiency in step 2, for example, in a form of electron stalling that results in electron leak or inefficient electron transfer promoting electron leak, that causes a reaction of these electrons to molecular oxygen forming superoxide O2 negative here. In skeletal muscle, there is an abundance of superoxide dismutase such that the O2 negative is then converted into H2O2. Chris Perry, who's in the audience, knows a lot about this. And of course, many of the people in the lab, presumably attending, will know about this too. So inefficiency in step 2 can cause H2O2 production / oxidative stress. And this has implication not just in skeletal muscle, but in other tissues. I think it shouldn't just be me that have heard the term oxidative stress in association with metabolic disorders. And I'll be talking about that a little bit later also in reference to skeletal muscle. Now, inefficiency in other systems would also be predicted to alter consequences to the cell. Inefficiency in ATP synthesis, so that's sort of a mix of between steps 3 and 4, would be predicted to probably be detrimental to cardiac muscle, because the inefficiency and mitochondria's activity to generate ATP will probably compromise contractility. Also, excessive generation of heat would cause malignant hypothermia in the heart. In pancreatic beta cells, inability for its mitochondria to effectively sense the difference in ADP and energy availability in the form of ADP to ATP ratio would also affect glucose-stimulated insulin secretion. And the stalling of substrates in step 1 and perhaps rather a differential stalling of different substrates stalling in step 1 is manifest in the form of perhaps insulin resistance or a change in substrate shifts that happen in the Wahlberg effect in cancer. So there are different consequences, and I know I'm going very slow here. I don't have that many slides, but I'll promise you latter slide I'll go through a lot more quickly. Hopefully you're not falling asleep, but for those who are mitochondrial enthusiasts, I think you find this reasonable and hopefully entertaining. But anyway, the point again to break down these steps is to think about and quantify efficiency in relations to what downstream consequences that such inefficiency would cause to the physiology. And when we say efficiency, we're not just sort of thinking about the activity. I think it's important to define efficiency separately from activity or capacity. Efficiency is neither of those things. Efficiency is a ratio between the energy that is coming out over the energy that has been invested or going into the system. And so we can quantify efficiency for each of these steps using different methods that we have developed and are developing, mainly by using high resolution spirometry and fluorometry that I've actually learned how to do in East Carolina from Daryl, who's Chris's former mentor. So that's what we are trying to do. We are trying to identify how efficiency of mitochondrial oxidative, inefficiency of mitochondrial oxidative phosphorylation could contribute to pathogenesis of metabolic disorders. And that's because, like I said, depending on which steps of the OXPHOS we were talking about, the consequences of low efficiency would be different. And we, as I have described just a little bit earlier, predict these consequences to contribute to different disorders, metabolic disorders in different diseases. And today I will be talking about our research on skeletal muscle mitochondrial inefficiencies shown on the left. But going one step further, so we're not the only one who is interested in studying what I've just described before. Chris may be one of them. But in fact, I think Chris is also interested in what I'm about to talk. So we're actually not even the only one that is studying what I'm about to explain also. But the step that we are interested in is really identifying the mechanisms that regulate the efficiency of these steps. And there are, like I said, many groups that study this, including the Mulyo, who recently had a paper on post-translational modification in the most recent case, acetylation of OXPHOS enzymes and the effect it has on efficiency. There are other groups that are interested in specific enzymes that may be regulating the efficiency of these enzymes. So both in the context of the transcription of these proteins as well as import of these proteins. We are interested in the role that the membrane lipid composition of intermitochondrial membrane plays on OXPHOS efficiency. And there are three reasons that we think that the intermitochondrial membrane is important. One is based on our empirical knowledge that the lipid composition of intermitochondrial membrane is unique. And it's not only just unique, but IMM is autonomously capable of altering IMM lipid composition. When I say IMM, I mean intermitochondrial membrane. So IMM is highly enriched particularly in two classes of phospholipids, cardiolipin shown here as CL and phosphatidylethylamine shown here as PE. Both of these lipids are cone-shaped, meaning that they are able to create membrane curvature. And as many of you would hopefully realize by me saying that the inner mitochondrial membrane indeed has great amount of curvature, especially in the cristae, both at the apex and at the cristae junction. And the important information that you may not be aware of here is that the enzymes who produces these lipids, PE and cardiolipin, are there in the inner mitochondrial membrane. These lipids are generated at the inner mitochondrial membrane for PE by an enzyme phosphatidylserine decarboxylase, or PSD, and for cardiolipin by a series of enzymes that produces cardiolipin. So this is one reason we think, just based on the fact that Imm lipids can change by machinery that's embedded in its environment. Second is that these lipids, the Imm lipids, are known to bind to enzymes of oxidative phosphorylation, and in many cases, absence of these lipids alter the activity of these OXPHOS enzymes. And these are shown in some cases by cryo-EM or crystallography, where the binding pockets for these lipids are identified and crystallized with or imaged, not necessarily imaged, or detected with these lipids bound already. And of note is that many of these lipids are found with these lipids, cardiolipin and PE, and also PC, bound to these enzymes already. So that's the second point, that we know that these lipids bind and affect the activity, but not necessarily efficiency of these enzymes. And a third point is that these lipids, PE and cardiolipin, we know that there are human mutations that affect the synthesis of these lipids that are classified as mitochondrial disease. And in both of these situations, the pathology is quite severe. Bar syndrome is a mutation in a tafazin gene. This is a final enzyme of the cardiolipin synthesis, and I'll talk about that at the end of my presentation today. But it's an excellent mutation, meaning that most people who have Bar syndromes are boys, although there is a report on female Bar syndrome, which would be the homozygous mutations to the tafazin gene. And last year, a couple group, one in Alberta and then another in Ireland, have reported mutations of the PISD gene, which is the enzyme that synthesizes PE. So in both of these situations, the phenotype is severe mitochondrial dysfunction, again supporting the idea that these lipids are important for mitochondria. But the question we're addressing is slightly different from that of Bar syndrome or the PISD deficiency, because the phenotype that we, as you will see, observe with metabolic adaptations, I will present data on skeletal muscle, but in other tissue, is not as severe as those we find in these genetic disorders. The changes in mitochondrial lipids are a little bit more modest. And so the questions we have is whether the change in these mitochondrial phospholipids that happen with metabolic interventions, we're talking about things like exercise and overeating, how does those interventions change skeletal muscle mitochondrial phospholipids, and then in turn, whether the changes in mitochondrial phospholipids themselves is sufficient to change efficiency of OXPHOS, as well as propensity for these metabolic disorders. Okay, so now getting into more data and talking exclusively about skeletal muscle mitochondria. So as an exercise physiologist, and for many exercise physiologists that are hopefully in the audience, I think we know from our training and also intuitively that this field have acknowledged about this concept of skeletal muscle inefficiency or efficiency for a very long time. And we know that the exercise can improve efficiency. And we're not just talking about mitochondrial efficiency. We're talking about efficiency of every energy conversion steps that promotes physical activity, exercise. So that includes things like the smoothness of joint and our pattern of our gait, to the change in myosin heavy chain isoform and whatnot. But the point here is that in our field, we have been aware about this concept that for every energy conversion steps, there always is an energy that leaks out. And of course, we all know this. This is a universal law, the law of thermodynamics, that the delta G does not equals delta H, but there is always an energy leaking out in the form of T delta S. And I'm giving a little shout out to Harmon Wasserman, who passed away a few months ago, who has really published a nice review coupled with data, an opinion piece that talks about this, including the concept of mitochondrial inefficiency. And so we know that the exercise can improve mitochondrial efficiency. And when we talk about that, we need to break it down in the forms of what I described in steps before. And on the top right corner is data on electron transfer efficiency shown by H2O2 production, which is analogous to percent electron leak here. And train TC controlled training reduces electron leak, meaning there's an increase in electron transfer efficiency. And on the bottom right is P2O, what it's often referred to as, stands for the ratio of ATP synthesis to oxygen consumed. And this value is increased in train, meaning there's more ATP produced per oxygen consumed, again, increase in the efficiency of ATP synthesis. So that P2O is more or less a combination of step 3 and step 4. I'd like to first focus my talk on the efficiency of electron transfer. And the reason why I want to focus on that is, well, for one, we know that increased electron leak is implicated in metabolic disorder. And specifically in skeletal muscle, there's actually at least a couple of negative phenotypes, so to speak, that is implicated with oxidative stress. One is skeletal muscle atrophy, so that is the loss of muscle mass. And the other is insulin resistance. Both of these happens with the lack of exercise or the lack of physical activity, inactivity or disuse. And in disuse or inactivity, there's a rise in electron leak that is known to happen in skeletal muscle. Similarly, inactivity also promotes insulin resistance, and this happens independent of changes in diet or adiposity. So inactivity in itself also causes insulin resistance, and greater electron leak has been implicated in pathogenesis of inactivity insulin resistance. So we were interested in the mechanism for increased electron leak induced by inactivity, and wanted to know if it happens because of the changes in mitochondrial phospholipids. So this study was led by Tim Heaton, a former postdoc in my lab, now a junior faculty in Minnesota. And when Tim first joined my lab, he did the very simple but good experiment where he exercise trained with treadmill mice over several weeks, and after which he dissected the muscle and then isolated mitochondria from these muscles to do a comprehensive lipidome of the mitochondria. And the data is shown here. I don't know if you can actually see the cursor. I'm assuming that you are able to see it, but the UT stands for untrained, T stands for trained on y-axis, And on x-axis are different classes of lipids shown on the top, PCP, CO, and whatnot. And each box indicate different species of such classes of lipids. And so you see different changes, variability along with it. We've also done the same experiment with a model of inactivity in Heinemann unloading. And you see something similar here. HU on y-axis stands for Heinemann loading. And so we have the mitochondrial lipidome from skeletal muscle in exercise-trained state as well as inactive state. And one of the common things that we found here was PE. So the yellow shows up and blue shows down. So exercise promotes the proportion of PE in mitochondria, and inactivity promotes the decrease in the proportion of PE in mitochondria. And along with that, there is an increase in the transcript of PSD, which is an enzyme that makes PE in a mitochondrial membrane. So this enzyme goes up with training, goes down with inactivity, which is what you would expect. So now we wanted to know whether changes in increased or decreased mitochondrial PE is sufficient to cause some of these changes in mitochondrial bioenergetics as well as the phenotype of skeletal muscle. So the first of these studies were led by Jordan, a PhD student that just defended his dissertation last week. And in part of one of his studies of his dissertation, he generated and studied skeletal muscle specific tamoxifen inducible knockout of this enzyme, knock-in of this enzyme PSD. So just briefly, this is a construct that would allow for a tamoxifen inducible skeletal muscle specific increase in PSD. And we show that here on the left, PSD transcript is elevated in the muscle, but not in other tissues. And the mitochondrial lipidome, specifically for PE, we see an increase in several species of PE utilizing this method. An important point to note is that this intervention did not change the mitochondrial content of skeletal muscle. So what happens to the skeletal muscle? It has greater level of mitochondrial PE, but the same level of mitochondrial enzymes. So utilizing the respirometry and fluorometry, we found that there is a significant increase in the capacity of oxphos quantified both by JO2 and JATP. So I think that is meaningful that the level of a PE, independent of the abundance of oxphos enzyme, can actually increase the capacity. So this is novel, but it's also somewhat consistent with what we may have predicted from some of the in vitro data that have been published by other groups. But what happens to the muscle of these knock-in mice? And unfortunately, this is the reason why Jordan didn't do this for his dissertation, not very much. There's not much that happens in these mice, at least in unstressed conditions. And we are stressing these conditions, as you will see later. I'll talk about that later. But there was no change in force generating capacity shown on the right, no change in fiber type, which is not that we would expect that. There's no change in vivo running capacity. So not a robust change. Is there some change that we might find if we probe the system further? Perhaps. We're looking into that in more details, but nothing obvious in these mice. But what about the other way around? So going back to Tim's data, this is the project that was funded by his F32 postdoctoral fellowship. And so he knocked out PSD using the same Cree mice that we got from Karen Esser. So again, this is a tamoxifen specific knockout of PSD. Knockout of PSD manifested and reduced transcript of PSD in gastrocnemius and diaphragm muscles, but not in other tissues. And low PE shown by TLC and mass spec. So what is the phenotype of the PSD MKO mice? Well, it was quite robust. So 2 weeks or 2 to 3 weeks after tamoxifen injection, so that's when the knockout is initiated. Knockout probably happens fairly quickly, but the downstream effectors will be predicted to take a little bit longer. The changes in mRNA, changes in protein, but also changes in phospholipid. It's not clear how long it takes for phospholipids to turn over, but it's predicted to take about 2 to 3 weeks at minimum. And so based on that information, almost immediately as the phospholipidome change, there is a decrease in body weight, a very rapid, quick decline in body weight, which is explained both by decline in lean mass and fat mass. And because of the lean mass, there's a kyphosis. When you open up these mice, there's the muscle atrophy. So I would like to remind you here again that one of the pathology indicated by increased electron leak is muscle atrophy, and that's what we see, quite robust atrophy. This is, again, 4 weeks after tamoxifen injection, so very short period of time, muscle mass is reduced. And not just muscle mass on the left, but also the force generating capacity of the muscle. This is an EDA muscle. It's substantially reduced in red. So much so that these mice die. And the reason why these mice die is because of the ventilatory failure. Diaphragm and other respiratory muscles are muscles, they're skeletal muscles. And so few weeks after these mice have begun to lose their body mass, they die. In between 6 to 8 weeks post-tamoxifen injection. At the very end, these mice have very low breathing rate and saturated oxygen in the circulation. And it's supported by the fact that the diaphragm mass, diaphragm force generating capacity are both reduced, and in the diaphragm muscle there is a fibrosis shown here in Cirrus red. So these muscles are bad. They are not contracting, their mass is reduced. What is the mechanism for that? Well, we tweaked the enzyme that changes mitochondrial lipidomes. So we looked into mitochondria. Left it is an image. Mitochondria seems bloated. Their cristae is not as tight. IMF stands for intermyofibular and SS stands for subsarcolemmal. So at both of these locations, mitochondria is larger, but not as dense in terms of cristae and electron density. And so they don't look normal. On the right is the respirometry and fluorometry. It's similar to what I've shown in for knock-in data. The ability of these mitochondria to consume oxygen or produce ATP is lower. So the capacity for ATP synthesis and oxygen consumption is lower. And again, this happens in the absence of the changes in mitochondrial enzymes. And shown here, even though the abundance is not different, the activities themselves for 1 through 4 is substantially reduced. But I would like to point out here, actually, that these changes that we see in morphology and changes in capacity for OXPHOS in it themselves don't explain the pathology. Definitely not death. We know that, for example, inactivity can reduce capacity to this extent. We don't predict rapid death in those type of conditions. In many different mitochondrial pathology whose defect is not as severe as what we've seen in this mice, we see the EM mitochondria like this. So even though this looks robustly defective, it doesn't explain the death. So what does? So that's what we've been trying to understand. And one clue that we have initially thought that would explain is the efficiency of electron transfer of the OXPHOS. And we think that the mechanism for this may be explained by the formation of supercomplexes of the electron transfer system, which explains that the efficiency of electron transfer may be increased because of these complexes forming a super or mega complexes that allow for a more tight transfer of electrons. And when such formation is absent, as you see here in the knockout mice, you would predict increased electron leak and oxidative stress. And that's indeed what we see. We see an increase in the ratio. This is the efficiency of step 2. Ratio of H2O2 over O2. In fact, the absolute H2O2 is also greater. We see it that manifested in greater 4-HNE, which is the lipid peroxide byproduct. Counter antioxidant mechanism being upregulated, and some of the apoptosis mechanism that's or markers that is being upregulated as a result of these ROS. So we said, okay, so maybe we have the mechanism. The deletion in PSD may be promoting oxidative stress to the extent it's triggering apoptosis. So to address that, we cross these mice with a mice that overexpresses mitochondrial targeted catalase. And I think some of you are familiar with this enzyme. The catalase is normally a peroxisomal localized enzyme that neutralizes H2O2 into O2. Into H2O. And so we have used the mutated form of this catalase that has a mitochondrial targeted sequence and called the MCAT, and we crossed that as shown here, and that could successfully negate the inefficiency in electron transfer shown here on the bottom right. However, overexpression of MCAT did nothing whatsoever to the phenotype of the mice. Physiology, whole body physiology shown here. No difference between red and pink lines for the loss in body weight, nor percent survival. If you look at this paper, there are data on O2 that's not rescued. The data on the muscle mass that's also not rescued. The forced genetic capacity also not rescued. So despite our hypothesis that oxidative stress may be causing the muscle atrophy and weakness in these mice, that was not the case. We followed it up with some hand waving transcriptomic data, which showed that consistent with the H2O2 data showing here, the glutathione and peroxisome metabolism is rescued with MCAT, but there are a whole bunch of things here shown here in the first two comparisons that are not rescued as a result of MCAT. So all these things, pathways, maybe are not rescued as a result of MCAT overexpression and may be contributing to the death phenotype or the atrophy phenotype. Some of them you see here, proteasome, ubiquitin-mediated proteolysis. I think lysosome may have been one of them. But so there are many potential downstream effectors that we're currently pursuing. But I'm gonna stop there for this. Homozygous mice have reduced mitochondrial PE, but probably greater than, well, not probably, but greater extent than what we saw in the high-limb unloading model of the mouse. And so we then reasoned that, okay, this is interesting. The homozygous knockout is interesting, but maybe it's not necessarily relevant to what we see in normal changes in mitochondria, mitochondrial lipids. And so Pia, PhD third-year student in our lab, has been working on this project. And that's because these mice, which we call a PSD MHET because they are heterozygous knockout instead of homozygous knockout, are more prone to insulin resistance, shown left as a insulin tolerance test, and on the right is the ex vivo 2-deoxyglucose uptake assay. And on the purple bar, you see that the insulin is unable to stimulate the glucose uptake to the same extent in control mice. And so this suggests skeletal muscle insulin resistance. So Pia is studying PSD MHET mice in the context of inactivity-induced insulin resistance, as well as the PSD MKI mice that Jordan generated. to see if that is protective from inactivity-induced insulin resistance. But there's one big technical hurdles to doing this, which Pia had to address, and it's a technical problem, but I think it's a very meaningful progress. So I'll explain what that is. And that is a problem with the lack of appropriate model for mouse inactivity to study insulin resistance. And many groups have shown that inactivity can promote skeletal muscle insulin resistance in humans. For humans, it's very simple. You ask them to voluntarily sit in a bed and don't move, and they will listen to you. Mice don't do that. You have to force inactivity in mice. You can't anesthetize mice forever, and besides, anesthetization is stressful. But there are many different models of inactivity, such as high limb unloading that I've talked before, high limb immobilization, denervation. All of these are highly stressful. And because insulin resistance is an endocrine disorder, we hesitated to use these highly stressful model of physical inactivity in mice. And in fact, as far as high limb unloading goes, it is absolutely a useless model for studying insulin resistance in muscle, because it actually increases 2-deoxyglucose uptake in the skeletal muscle. So Pia and Ziad is a postdoc in Micah Drummond's lab, who we are a co-PI on this, have been studying and exploring different models for physical inactivity to study metabolism. And the ones that we have arrived to, which is not novel in a concept, but novel in the design, is a small cage housing. So small cage housing has been used by other groups, many other groups, rats also, which happens to be a better model to study inactivity and insulin resistance than mice. But rats have limited availability for genetic studies, and we don't have genetic rat models, so we needed a mouse model for inactivity. And small cage housing, Pia and Ziad, this is the third generation design of our small cage housing, single cage housing for mouse, that produces an ideal, I think, a phenotype to study inactivity and insulin resistance. And one of the reasons why I call it ideal is because this intervention does not change the adiposity shown here in a fat mass by NMR and weighing of the individual fat pads, and this is epididymal fat. Small cage housing does not alter adiposity, and it causes insulin resistance shown here, demonstrated by the reduction in insulin-stimulated 2-deoxyglucose uptake. So thanks to Pia and Ziad, we now have a great model to study insulin resistance in mice induced by physical inactivity, and Pia is using this particular model to study inactivity. Now, so that's all I have for this topic. Now I'm gonna switch gear for the last 5 or so minutes. I don't wanna go over too much time, but this is on a completely different project. So we have been so far talking about step 2 efficiency. Now we're gonna switch gear to a project on step 3/4 efficiency. And the reason why we came to be interested in this is on this concept of energy efficiency, or the efficiency or ATP synthesis in skeletal muscle that may be relevant in weight loss. And so a group led by Rudy Leibel has been studying this for a while. And the idea is when a person loses weight, one of the reasons why these individuals may be more prone to gain their weight back is because their energy efficiency is higher. That the weight loss induces increasing energy efficiency and decreased energy expenditure. And that's supported by these data here. This is a longitudinal study with weight loss. So there's an energy expenditure before weight loss and after weight loss on the right, 10% weight loss, and 24 hours energy expenditure total or normalized to fat-free mass is lower in weight loss state. And interestingly, the non-resting energy expenditure, so that's energy expenditure that is above what's expected of the basal state, as in mostly exercise. And in this case, as you all would agree, most of the energy expenditure that is induced by physical activity comes from skeletal muscle. And this is reduced, suggesting that skeletal muscle energy expenditure may be lower after weight loss. So Pat, a PhD student that had graduated from my lab, now looking for a postdoc, but because of COVID, that process ended, and he's doing a brief postdoc with Micah, just so that he can diversify his skill sets rather than sticking around in my lab. I commend him for that. Along with Marissa, a newer PhD student, she's a second-year student, is working on this project, have modeled weight loss in the mice. And this was a simple study where wild-type C57 Black 6J mice were placed on a high-fat diet feeding regimen over 10 weeks. And after 10 weeks, by the way, these are single house, half of the mice were kept on the high-fat diet, and the rest, the other half, were switched to standard chow, so non-high-fat diet. And what you see here is interesting. There is a weight loss in the first couple weeks, and then the weight stabilizes. And I think that's an important point here. There was no longer a situation where mice were actively losing weight anymore. So the weight was stable. Lost, still above the weight of the lean mice or the mice that were fed standard chow only, but somewhere in between. And consistent with Rudy's findings, we found the whole body energy expenditure to be reduced. This is normalized to lean mass, is reduced in the weight loss group. Not so surprising, just consistent. But what is new about their findings, our finding, is the efficiency of skeletal muscle energy expenditure. And so this can be quantified by ATP produced per oxygen consumed. Step 3-4 efficiency is what we call it. And this is higher. And, you know, I think in the world where we often find papers studying genetically modified models, we become kind of numb to seeing 2 to 3-fold increase. I think this change that we see approximately 50% increase in wild type mice with this weight loss intervention is substantial. That there is a 40 to 50% increase in P to O ratio in wild type mice after weight loss. So we think this is hugely impactful to the physiology of the skeletal muscle and the system physiology. And I have just a couple more slides. What we think is important for this mechanism for increased P2O or reduced energy expenditure in these mice is this molecule, a tetralinoleocardiolipin, which is one species of cardiolipin with 4 acyl chains that are all 18-2 linoleic acid. So tetralinoleocardiolipin or TLCO. This is increased by 2-fold in a wild type mice, wild type weight loss mice compared to obese. And the reason why we're interested is going back to earlier slide where I was talking about Bart's syndrome. Bart's syndrome is a defect in an enzyme of the terminal enzyme of cardiolipin synthesis, tafazin. And by having a defective tafazin, this molecule, TLCO, is not being generated. It is, in fact, not just TLCO, total cardiolipin goes down. But this is an enzyme that's being defective. And in the paper we published a couple years ago, this is what we found. This was not the focus of our study. That paper that came out in JMCC focused on the cardiomyopathy induced by Bart's syndrome in this mouse model. But what we found was we wanted to study these mouse over many months of time because we wanted to know the progression of cardiomyopathy. And what we stumbled across, this is not a high-fat diet. But what we found was the fat mass of the tafazin knockdown mice, so this is the model for Bart's syndrome, was much lower compared to the wild-type mice. And this was explained, again, by an increase in energy expenditure and not by differences in food consumption. So we think that the TLCL regulates mitochondrial efficiency for ATP synthesis, and this change that can happen in Bar syndrome and in tafazin knockdown mice is sufficient to change whole body energy expenditure. So we are actively pursuing the role that TLCO and other cardiolipin molecules have on the energy expenditure and efficiency of the ATP synthesis. So this is my last slide. Just going back to, just generally, we talked about the muscle phenotype today, the atrophy and insulin resistance, and now energy expenditure of a skeletal muscle that is affected by mitochondrial lipids. We are also interested in many other tissues and different pathologies that are potentially caused by changes in mitochondrial lipids and efficiency. So, and by that, I'd like to acknowledge those that are involved. Those that are highlighted in red are specifically for people that are involved in the skeletal muscle project in my lab. Pat, Jordan, Marissa, Pia, Tim, and Tony have been heavily invested and involved in these projects. And acknowledge Micah, who's a collaborator at Utah, heavily involved. Daryl and Kelsey, also a faculty at ECU, formerly a PhD student with Daryl. I think Chris knows him fairly well too, and a few other collaborators. And I'd like to take questions. Sorry for going over a little bit.
Well, Katsu, thank you very much for that. Very nice. You know, it's not often we talk about metabolic efficiency. We talk a lot about mitochondrial content and proteins and things like that. And I think your talk has nicely shown us that we have more things to study about mitochondria related to efficiency and that sort of thing. Very nice information. So we're a little over, but let's take a bit of time for questions through the Q&A, folks, okay? If you don't mind, ask a question through the Q&A. I'll read the question for Katsu, and then he can have a chance to respond. So Chris is asking, Chris Perry, did you measure mitochondrial respiration using pyruvate or a different substrate? There's evidence that activating pyruvate dehydrogenase could treat Barth syndrome.
So are you referring to our PSD part of the talk or the tafazin side of the talk?
I think, you know, the last part of the talk.
Okay, okay. So the tafazin one, we actually haven't done a lot of the respirometry. What you saw at the end with tafazin knockdown, that was a whole body VO2, not JO2. We did do JO2 in that paper. We weren't paying that much attention because we were, I mean, we did do JH2O2 there also. I think it was just with pyruvate that we did, not just with pyruvate, we didn't do it with succinate as an alternative substrate. I think we did it with pyruvate malate. I don't remember if glutamate was in there or not. But this is a good point because, well, depends on what exactly Chris is alluding to. What you said is activating pyruvate dehydrogenase could treat Barth syndrome. So the idea is that, what is the idea? The idea is that
just asking you what substrate
cause a defect in pyruvate oxidation. Is that the implied defect here? And if that's the defect, I would actually agree with this. We have some hypothesis on this that I guess I could share this with you. Is the differential effect, and I don't know if it's the defect is pyruvate dehydrogenase. What we are actually trying to pay attention is the defect is on the mitochondrial pyruvate carrier. And this is a reason. So if you change cardiolipin, which happens with Barth syndrome, the first site, the obvious site to look at is the inner-mitochondrial membrane, right? And so if you, and we're not looking at this just from the context of pyruvate oxidation, but actually sort of this concept of insulin resistance and what would more likely to be affected in terms of substrate preference. So cardiolipin is more enriched, highly enriched in inner membrane, less so, but do exist in outer mitochondrial membrane. We know that the rate limiting step of pyruvate oxidation is mitochondrial, well, that could be actually argued. But we know that the mitochondrial pyruvate carrier is essential for pyruvate oxidation by means of pyruvate import into mitochondria. And this is localized in Imm. We know, on the other hand, that the fatty acid oxidation, its rate limiting step, I would also actually argue with this, but the dogma is that the CPT1, carnitine palmitoyl transferase 1, is the rate limiting step. CPT1 is localized in OMM. MPC is localized in IMM. So if you have a defect in cardiolipin, you would expect the defect to be more preferentially on the pyruvate side and not as much on the fatty acid side. We do have some data on this, but not like very good. We are working on that. So I do agree with this notion that the cardiolipin, or rather the lack thereof, may preferentially affect pyruvate oxidation. And if the thought is that you could rescue Barr syndrome's defect with pyruvate, I think there is some merit to that.
Really remains to be determined.
Yeah, but you know, I'm actually a little bit skeptical about, please don't advertise this much, but I'm kind of skeptical about the mitochondrial origin of Barr syndrome, which is kind of weird things to say. But there are evidence that suggests that the cardiolipin can actually affect the genomic transcription. And I used to not have a strong belief on that, but I now believe that a lot more strongly. And I think that is something worth paying attention to, even though cardiolipin is rarely extra mitochondrial. Yeah. And I think there's something to be said there. I have no idea what the mechanism is.
Well, Katsu, there's I got a few more questions here for you.
Okay.
Related to your last point, I mean, there's definitely some retrograde signaling from the mitochondria and to the nucleus, right? So your point of you know, how something in the mitochondria and cardiolipin, for example, might regulate gene expression is certainly a good point to make. So let me get you to answer a couple more. Let me ask the people outside to please use the Q&A option on the bottom of their thing to ask the questions. Rolando Sedia is asking you, how do you explain the reduction in fat mass in the PSD muscle-specific knockout mice?
Yeah, that's simply because they're not eating, because they are not mobile. I mean, they have reduced food consumption, and we think that's the reason for reduction in fat mass. They just don't look good. They look sick. They don't move around, and I think I don't know if it's a central drive or not, but it's simply they just can't eat as well because they're weak. That's our interpretation.
That makes sense. So Dan Lark is asking saying that he found the P to O ratio doesn't change with voluntary wheel running in chow-fed mice. Do you think the high-fat diet is required to for the changes in that cardiolipin, TLCL, and the P to O ratio?
Yeah, you know, this is really interesting. The notion that exercise changes P to O is actually not always supported by data. I've shown the data that supports what I'm trying to say, and that's what you show, you've seen. But yeah, it's kind of questionable. Why is it about the exercise that do this? It's certainly the case that the regular exercise seems to not be sufficient, that there needs to be a adaptive mechanism in place. And so it needs to be a training. It needs to be, I think, a pretty laborious training. With respect to the what's causing the changes in P to O or TLCL for that matter with weight loss, you know, we're just beginning to address this. Is it diet dependent? It's difficult to say. I have to go back and look at we've done the high-fat diet-induced changes in muscle lipidome also. I don't remember this, and the reason why I don't remember is because we are thinking that maybe the changes in P2O is happening happen because the weight-bearing nature of the skeletal muscle. When you lose weight, you lose the weight bearing of the muscle, and so we think this may have more intrinsic origin to the energetics of the muscle rather than the lipids that's coming in. That's not based on data yet. We have some anecdotal data to support this notion, but it could be because in the obese state, the muscle adapted to carry the increase in body weight by making an appropriate adaptation, and then you have this rapid weight loss. All of a sudden, as you saw in our data, the weight changes in 1 or 2 weeks, and then so there's this, not a crisis, but there's this lack of adaptation that happens because the weight changed all of a sudden, and the muscle is not carrying as much weight anymore. So that's what we're thinking, and that will be kind of consistent with data in humans that the weight loss that happens acutely is less likely successful to help people have their weight loss. And the longer the person is able to maintain their weight, less likely they're going to bounce back. But there's all kinds of yellow flag with that statement because there's a lot of drive that's involved in appetite regulation.
Right.
And
what about the dietary induced? Chris McGlory is asking, do you think the dietary induced manipulation of lipid composition of the mitochondrial membrane influences some of your outcome? For example, ingestion of omega-6 versus omega-3 fatty acids, that kind of thing?
Yeah, it's an interesting point that is very difficult to address. You would agree, Chris, because by nature, the entry of these lipids is oral and it goes everywhere, right? It goes all kinds of tissue and it changes a whole bunch of other things, not just mitochondrial lipids. And there are certainly implications that a diet-induced changes in mitochondrial lipids would have, but it's very difficult to study this. I don't know if that answers your questions. In terms of omega-3 and omega-6, yes, I think it's important, but I think the question is, what is it important for? And is it important for atrophy, insulin resistance, this P2O? I think the answer is not the same for these three. With respect to omega-3, 18.2 is omega-6. So, yeah, maybe. Maybe.
So
it's complicated.
What about the change in composition affecting carbohydrate or lipid oxidation? Chris Perry is asking. Does it, changing the lipid composition, does it affect fat metabolism versus carbohydrate metabolism? Or do we know that?
Yeah. So, I mean, so in the case of PE, that's what it implies, right? That if you reduced PE, you have reduced insulin-stimulated glucose uptake in the skeletal muscle. In the level of respirometry, I don't think we compared. We are going to do that, or PIA is going to do that head to head, from the mitochondria isolated from the same muscle, comparing. What we're doing is comparing like palmitoyl-CoA plus carnitine with palmitoyl-carnitine. So that's sort of like a pre-receptor, post-receptor idea, substrate-dependent oxidation for CPT1. And for pyruvate, we're doing pyruvate and pyruvate ester. So again, the pre to post kind of substrate-dependent oxidation, and compare that to see if we're going to have some sort of a preferential defect as a result of the lack of PE or cardiolipin, whatnot. Also mixed substrate with including the CPT1 inhibitor or MPC inhibitor. So we're doing those experiments. We don't have an answer. Certainly, we think that there will be a difference based on what I've described before, with the fact that that the MPC is localized in IMM when there's a lot of cardiolipin, and CP21 is localized in OMM when there's a lot less cardiolipin, or PE, same thing.
Okay. So in one of your last slides, you showed the weight loss, and Daniel is asking, there was almost an instant decrease in body weight in the weight loss group, and he's questioning whether the food intake was different between the two groups, and if not, how did the diet composition of the high-fat and standard chow diets contribute to the difference in metabolic efficiency?
Yeah, I can't answer about the composition. I haven't looked at that carefully. As far as the food intake, certainly the caloric intake is lower. I don't think that's why you're asking though, because we have a different, high-fat diet is more calorie dense. So total caloric intake is definitely lower when you switch to standard chow, and we think that's why they lost weight. As far as the total gram of chow, standard or high-fat consumed, I don't know. We have the data. I don't know. Don't know.
One of the last questions here, Katsu. You've been on alert for a while here, so we'll give you a break. Mohammed is asking, are there any data that suggest that exercise-induced glucose uptake is impacted the same way that insulin-mediated glucose uptake is impacted in the knockout PSD muscle-specific knockout mice?
That's a loaded question because there are multiple effects of exercise on glucose uptake, right? There's an acute effect of exercise on insulin-independent increase in glucose uptake, which I think you might be referring to. There's also acute effect of exercise on insulin-stimulated glucose uptake, which is referred to as exercise-induced increase in insulin sensitivity. And there's also a chronic effect of exercise on insulin responsiveness. And then there's a few other things. But so if you're referring to the effect of exercise on insulin-independent glucose uptake, in this particular case, I don't have data, but I think it would not be affected because I don't think this is, well, actually, that might be wrong. But I think it's probably reasonable to say that it's a lot less likely to be affected because the mechanism presumably is exercise stimulates AMPK, but that's maybe mitochondria dependent, and that promotes GLUT4 translocation. Yeah, I don't know.
Fine. You know what? It's fine that you don't know. There's a lot of things I don't know. So, it's okay. We're good. Does anybody else have any more questions for Katsu? You know what? I want to thank you very much. You've taken a lot of questions and you gave us a lot of information, and certainly the area of mitochondrial lipids and the role in metabolic efficiency is an understudied area, and that's probably why you're doing so well funding-wise and things like that, because it's an area that really needs more attention. So I commend you on that, and I want to thank you very much for being with us today and answering a lot of questions and giving us some great information.
Thank you, David, and thanks for everyone for listening in. Great questions. And if you have any other questions, feel free to email me also.
Very good. So I'll ask you to stop sharing your slide. You can hang out for a minute, Katsu, and I'll just tell folks that the seminar is now over. And thank you very much for coming. And we'll see you actually next week. We have another seminar.
Thank you.
All right. Stop recording.